Yep, my name's Dylan. I'm a PhD student from the Marine Parasitology Lab at UQ, the same lab as Clarice. And just before I start, I'd like to acknowledge the traditional landowners of where we meet today and where all my research has been done, and pay my respects to the elders past, present, and emerging. So much like Clarice, I work on the life cycles of trematodes, but a completely different group. So I work on a family of trematodes called the Spirorchidae. Now, these are turtle parasites which infect the cardiovascular system of their hosts. So they're in the superfamily Schistostomatoidea, which includes the Aporocotylidea, which infect fishes, sharks, and rays, the Schistostomatidae, which infect mammals, birds, and crocodiles, and then the Spirorchidae, which infect turtles. Now, a lot of my work is just focused on marine turtles, so that's what I'm going to be talking to you about today. So of the 7 known species of marine turtles around the world, 5 are known hosts. So the only 2 that aren't known hosts for spirochids are the flatback and the leatherback, and it's likely that they're because they're quite rare and difficult to get samples from. So this is in terms of trematodes, it's a pretty diverse family. It consists of 33 species distributed across 10 genera, and as you can see on this slide, they come in a variety of different shapes, sizes, and they infect a lot of different locations within the host. They are quite pathogenic, these species. And they can occur at quite a high prevalence. So close to home here in Moreton Bay, just outside of Brisbane, they can occur at upwards of 75% prevalence. And of the turtles that do die in Moreton Bay, around 40% of the mortalities can be attributed to these parasites. So they're quite pathogenic. I'm not going to get into how these parasites cause disease today, because it's a little bit out of topic of what I'm talking about, but they do cause quite a bit of disease. What I'm really interested in is how they're getting around. So we just had that great talk from Clarice explaining the life cycle, so I'm not going to bore you with how they get around. But what I'm lucky with my group is that they have quite a simple life cycle compared to the Hemiuroidea. So spirochids have just a 2-host life cycle. So you've got the adult in the cardiovascular system. It lays the egg, which hatches into that larval stage, which goes on to infect an intermediate host. I'll talk more about the identity of this intermediate host in a second. Develops into that asexual adult that produces cercariae that merge into the water column and then go on to infect the turtle. It's a lot less complex than Clarice's system, which makes it a lot easier for me to work on. So we know a little bit about how these parasites are getting around, but a lot of the work that we do know comes from the freshwater realm. Now, freshwater systems are a little bit easier to work on than the marine realm, and that's essentially because if you have a sick turtle in a lake, you know that that intermediate host has to be in the lake somewhere, so it's just a matter of finding it. So we know that freshwater spirochids are using planorbid and ampullary snails, but unfortunately these groups of snails aren't in the marine realm, so it's not much help to me. Now we know a little bit about what's happening in the marine realm. So we've got preliminary work from 2010 from authors in America who suggested that keyhole limpets were an intermediate host for a species of spirochid called Leiridis leiridii. Now, what's important about this finding is that the authors didn't actually see any intermediate stages. They used PCR to get a positive parasite amplification from host tissue. And the case kind of went quiet for a few years until my supervisors and some collaborators from Spain identified vermeted gastropods as intermediate hosts in a turtle rehabilitation center. So they found the larval stages inside the vermeted gastropods and used molecular markers to match them to eggs on the gut of neonate turtles, as well as to an adult belonging to the genus Amphiochus infecting loggerheads. So we thought that was, pretty straightforward story. All spirochids seem to be using gastropods, nice and easy, we know where to look. And then a year later, a group from America identified Terebellid polychaetes as a intermediate host for another genus of marine spirochid known as Neospirochus, which kind of threw a spanner in the works. But when we have a look at how the intermediate host exploitation matches up with the phylogeny of the family, it makes a little bit of sense. As Amphiochus, which infects vermeted gastropods, is quite distantly related to Neospirochus, so much so that they might actually represent 2 families. So the Spirorchidae right now is paraphyletic, assuming that the Schistostomatidae is a valid family, which we think it is. So that's a little bit of explanation of maybe why we're seeing that host switch, but we don't really know much right now. Like I said, all that we do know is Amphiochus is definitely using vermeted gastropods, Neospirochus is using Terebellid polychaetes, and maybe Leiridis is using keyhole limpets. So this is where I've come in. I've gone around Australia at various locations sampling vermeted gastropods and Terebellid polychaetes. I've been looking at Moreton Bay, Heron Island, Lizard Island, and in captivity in SeaWorld. And because the larval stages don't look anything like their adult counterparts, I've been relying on molecular markers to identify species that have been found. So I've been using ITS2 and COX1 for species identification and 28S for any taxonomic inferences. What I've found so far in terms of vermeted gastropods as intermediate hosts, it varies at different locations. So starting off at Heron, it was a pretty fruitful expedition. So we found that the 4 species of Apalatrema are using vermeted gastropods at Heron Island, as well as the additional 3 unknown species. This is a really important finding from a conservation point of view, because Heron Island is an important turtle rookery. So essentially, we're showing that transmission's occurring at this island, and it has some implications on how the parasites are being spread, potentially to neonate turtles as they're entering the water just at hatching season. From a biodiversity point of view, this is also a really exciting finding, as we've got the 3 unidentified species, which essentially tell us that there is undescribed spirochete biodiversity in Australia, and then by extension elsewhere in the world. So these 3 unidentified species don't match any sequence data that's available online, and they, until recently, didn't match any sequence data that I generated from adults that I collected from deceased turtles. Moving on to Moreton Bay, we've got 3 of the 7 species we found at Heron Island at Moreton Bay. That's 2 species of Apalatrema and an additional unidentified species. Not overly surprising that we found these here, given that Moreton Bay is a well-known turtle foraging ground, and there's lots of vermeted gastropods there, so we expect transmission to occur, and it is. And then at Lizard Island, we found another species, Apalatrema post-orchids, occurring there. Again, not overly surprising, given that this is a green sea turtle parasite, and there's a lot of green sea turtles that inhabit the waters around Lizard Island. So at SeaWorld, we found 2 species. The first one is a perfect match to Amphiorchid, which was the larval stage identified from Valencia, Spain, sorry. And it was using the exact same intermediate host as well. So essentially what we're showing here is that the parasite and its intermediate host very likely have a circumglobal distribution, at least in captivity, which is really important, again, from a turtle conservation point of view, as these are clearly spreading in captivity and cause quite a bit of disease. From obviously a biodiversity point of view, it's really exciting that we're showing that this parasite is using an invasive species as the host circumglobally. Then the second species we found is a pretty close match to Liridius liridii. There's a little bit of divergence in ITS2, COX1, and 28S, but not enough to convince us that it's a different species. So we think we're seeing a little bit of geographic variation there. But if you remember, Liridius liridii is that species that previous authors had suggested was potentially using keyhole limpids as an intermediate host, but we found it in a vermeted gastropod. So there's something happening there that needs to be disentangled. And then jumping onto the Terebellid polychaetes as intermediate host, it's a lot less fruitful than the vermeted gastropods, unfortunately. Just found a single infection. All of that kind of cream stuff spilling out of that worm are all those larval stages, and there's one stained. Unfortunately, it's an immature infection, so we can't do any morphological characterization. But we can sequence it, and it looks to be a pretty close match to an unknown adult species from Florida, infecting green sea turtles. It's essentially showing that this parasite also has likely circumglobal distribution, as we found this in Moreton Bay, and the adult has been sequenced from Florida. To recap, what we knew before I started was Amphiorchid using vermeted gastropods, Neospirochus is using Terebellid polychaete, and we thought maybe Liridius was using keyhole limpids. But what I've essentially shown is that this entire clade is consistently using vermeted gastropods as intermediate hosts. And given that Liridius is nested right in that clade, and I've shown that it's using vermeted gastropods as intermediate hosts, the finding of a keyhole limpid as an intermediate host seems unlikely. What I think likely happened in that earlier study was potentially a vermeted gastropod was actually encrusting on the keyhole limpid and led to that positive PCR result, but it's hard to know without actually going back there and resampling again. We've also further shown or supported that Neospirochus seems to consistently infect Terebellid polychaetes, and with the substantial sequencing that I've done and assessment of the vermeted gastropod fauna along the east coast of Queensland, the fact that no Neospirochus has shown up in vermeted gastropods is really cementing our idea that these two have a very distinct intermediate host switch. Then on top of that, I guess the future work that we need to do is try and figure out what this little clade called Keratocola is doing. And the reason we're interested in what Corredicola is doing is because, like I said, we know what this clade is doing. We know that this little blue clade is infecting planorbid snails, the little freshwater clade. We know that top clade up the top is infecting mostly freshwater snails because these Cystomatidae. We know that that big blue clade at the bottom is also mostly freshwater with that little red clade in there, and they're all infecting freshwater snails as well. But Corredicola is the last marine clade that hasn't had anything really figured out for it. And the reason that we think this is interesting is understanding what Corredicola is using as an intermediate host will kind of guide our understanding of intermediate host exploitation by the Spirochaetae, and then also by extension, the superfamily blood flukes. What I mean by this is that if Corredicola is not using Vermeida gastropods as an intermediate host, we would think that the jump to Vermeida gastropods occurs of that orange arrow, and potentially that the green clade and Corredicola independently jumped into the marine realm as well, given that freshwater seems to be the plesiomorphic state for this group. Now, if Corredicola is using Vermeida gastropods as an intermediate host, we think that the jump would most likely occur at the purple arrow, and then that little blue clade itself would have independently jumped back into the freshwater realm, and that the common ancestor for that whole clade would have been marine. But because we don't really know what's happening with Corredicola, it's hard to make any judgment calls at the moment. And even when we do find it out, it's still going to be quite a complicated story to figure out the pattern of intermediate host exploitation by Spirochaetae and then by the Schistostomatoidea. So that's me. I just want to say thank you to all my funding bodies and all the collaborators down the bottom, and I'm happy to take any questions.
Awesome, thank you. Was it Dylan?
Yes.
No, sorry. So I have to apologize for that first up. You're okay on our program as Richard, not Dylan.
It says it on here as well.
Yeah, so I was so confused. Has anyone got any questions? All right, let's have a look. Nope. I am so out of my depth here, but I kind of like agree with Jeremy's comment. It blows my mind with this stuff that you kind of have to work backwards. It's like this full detective kind of thing, which I was looking at your phylogeny and just thinking, oh my God, there is so much work in that and so much sampling. Have you had to go and collect all of those samples yourself, or how much of how? Wow.
So I think a total, there's a few Vermeida gastropods that were examined prior to my PhD. But I think throughout in the last 2 years, I've done over 3,500 to 4,000 snails.
Wow.
Yeah. So it's been a lot of work, but it's fine.
Yeah, that is. And sorry if I missed this in your talk, but is that, was it all around Queensland?
Yes, it's all around Queensland.
Yeah. Awesome. Yeah, that just, I saw that and thought, wow, that is such a lot of lab work. Yeah, awesome. Awesome project. What's the time? Okay, if no one else has got any questions. Oh, Erin's got her hand up. Erin?
Yeah, Dylan, I was wondering, how do you actually find them in the snails when you collect them? Do you just do you sequence the whole thing, and can you pick up the trematode sequence, or do you actually dissect them out, or how does it work?
Yeah, so we dissect out every snail that we collect. So we kind of break the shell open, and then they've got the reproductive end of the snail, and the trematodes essentially take over the reproductive digestive gland. So when you open up the snail, you can instantly tell that it's infected or it's not, and then just kind of separate it from host tissue and store it and sequence it later.